Hello, everybody. Welcome back to Creation Myths. Just let me know in the chat, as I always like to check, if the sound is okay, and if there is an issue, just let me know so I can address it before I do a whole stream with nobody listening to me. So just let me know if there's any issues. Otherwise, I'm just going to keep going. Hi, everybody. Welcome. It's great that you're all here. I appreciate you all being here for this first installment in a new weekly series that I am planning to do. Fingers crossed. My thinking is that this is going to be a pretty straightforward series to do what I hope will be relatively short episodes each week. And what we are going to do here is talk about a series of books from Answers in Genesis called the New Answers books. There are 4 books in this series, and we are going to go through them chapter by chapter and talk about all the problems in these books. That's what we're going to do in this series. So this is part 1 of this series. This is the first episode of a series that will span potentially over 100, hopefully relatively short episodes. We're just going to do chapter 1 here. We've got the New Answers book 1. Chapter 1, we're going to go through it. We're going to pick out the errors and see what the problems are. I bought the Kindle editions of all 4 of these books. So I got all 4 of them. I'm all ready to go. We're ready to do this. And what we're going to do is we're just going to go through the text of it. I've highlighted places that I want to talk about. You can actually see my notes right here on screen. I don't know if you can see my cursor on there. I guess not. But you can see my notes on screen right there. And that's what we're going to talk about. So let's get right into it. We're just going to go. This book, it starts off, there's my cursor. Cool. So this book, there's information, acknowledgments, and special thanks. We've got our table of contents, and then we just get right into it. There's how many chapters are in this one? There are 27 plus a bonus chapter. So we'll cover most of these. The way I'm going to do this is I'm only going to cover the stuff that I'm qualified to comment on. So anything theology, geology, astronomy, any of that stuff, I'm going to skip. But if there's anything biology that I can comment on in a given chapter, I'll do an installment on that particular chapter, right? Now, that's how it's going to work for all 4 of these books. I don't know exactly what fraction of chapters are going to be relevant to my expertise. I've only read a few chapters in this first book of the 4. But that's what we're going to do. We're going to go through and just look and see if the arguments and the answers provided by Answers in Genesis, and specifically, you can see some of the authors here, so it actually varies by chapter. We could talk about that briefly. So you've got Ken Ham and Jason Lyle, Mortensen, Ham, Lyle, Ham, Riddle, just a whole bunch of different people. Michael Ord is another one, Bodie Hodge, Andrew Snelling. I think I saw Georgia Purdom on here. Yep, there's Georgia Purdom. So a bunch of different people, and we're just going to see if the arguments they make and like the factual basis for those arguments stands up to scrutiny. So after the table of contents, we're just bam, there's no introduction, there's no author's note, there's no nothing. We're just right into it. So our first chapter is there really a God? And you may be thinking, Dan, you just said you were only going to cover biology. This chapter might not be all that relevant. Turns out there's a lot of biology in this chapter, and it's basically all wrong. So we are going to go through it. So I'm going to skip parts. If there's any page that doesn't have highlighting on it, that's because there was nothing on that page that I wanted to talk about. There's nothing relevant there for me to comment on. So we're just going to skip right to the part where they talk about identifying biological design, but you can probably guess where this is going, because you can see here we've got good old Mount Rushmore, right? So we're talking about identifying the hallmarks of design in nature. So here's the first kind of big idea, right? Living things show evidence of design, and we're going to spend the rest of this chapter basically supporting that assertion. So let's do that. Let's see how they do that. You can probably guess some of the arguments that we're going to see here. Okay, first thing I'll just point out is, wouldn't it be logical to assume that if man's highly intelligent brain designed the computer, then the human brain was also the product of design? So this is a, wow, we're complicated, therefore we have to be designed kind of argument. And like, you need a basis for identifying design. You can't just be like, looks designed, therefore it's designed, or looks complicated, therefore it's designed. That's not a testable hypothesis. That's not a basis for assigning design as an explanation. You need an actual objective standard that you can use. And we know of a couple of such standards that creationists employ, and we'll see which ones or one they use here. So let's keep going. They're going to talk about the designer. This stuff doesn't matter. There's a lot of quotes of Dawkins. It doesn't matter, because what we really want to talk about is the biology. So they get into, does natural selection produce design? And in this section, we get a trick that creationists do all the time, which is break up mechanisms of evolution into each little one individually and say, well, this mechanism can't do X, and this mechanism on its own can't do X, and this mechanism can't do X. Therefore, evolution can't do X, ignoring the fact that all of these mechanisms are operating together with each other. So here's what we see. We say, therefore, to argue that natural selection and mutations are the basic mechanisms of the evolutionary process, one must show that these processes produce the information responsible for the design that is evident in living things. If you've watched my stuff, you know right away what a problem is here. Natural selection and mutation are not the only mechanisms of evolution. There are a ton of other mechanisms of evolution. And if you're going to limit your discussion to it's all just natural selection acting on mutation, then you're leaving out a whole bunch of how evolution works and a whole bunch of mechanisms that generate evolutionary change and evolutionary novelty. Okay, so right away we've got this kind of tunnel vision with a very old version of evolutionary theory. Then we get a gem, a real classic here, right? Natural selection only operates on the information that is already contained. It does not produce new information. Yeah, that's right. Natural selection doesn't generate anything new. It only operates on what's there. That's true. Other mechanisms generate new things through various means. 100% true. Yep. Let's keep going. By the way, this is sprinkled with like the silly cartoons that we know and love from AIG. So you'll see these just throughout. They're terrible. They're all through here. All right. So let's keep going here. I like this one. One kind has never been observed to change into a totally different kind with information that previously did not exist. Now you can see my note here, right here. You get a little look into my brain as we're doing this because my notes are going to be on screen, which is cool. I like that. So we have observed things changing, doing by what any reasonable standard is a word that I don't care for, but creationists like, and that word is macroevolution. Some directly observed examples of this are the ongoing primary endosymbiosis occurring in a member of the genus Pollinella that's converting a heterotrophic rhizarian. It's an amoeba-like thing. It's not technically an amoeba, but it's like an amoeba. And it's, so you've got this genus that is, I think, 7 species. 5 of them are heterotrophs, and 2 are photoautotrophs due to a relatively recent, it's millions of years, but in geological and evolutionary time, it's recent primary endosymbiosis, and it's a currently ongoing process right now that we're observing in the present, right? So you've got an amoeba-like thing becoming a completely new type of green algae before our eyes. That's macroevolution, my friends. That's definitely it. Another example here is Chlamydomonas. These are the green algae that, for their entire evolutionary history, have been unicellular, and in the lab, we've evolved obligate multicellular versions of these green algae. So it's going from unicellular to multicellular. By any reasonable standard, that's macroevolution. That's a new kind. And then the last thing on the list here, Elysia chlorotica is an animal that's actually undergoing endosymbiosis and becoming photosynthetic. It actually has undergone a process called nuclear gene transfer, where the animal genome in the nucleus of the animal cells has acquired genes from its algal food for photosynthesis. And it expresses some of the genes necessary for photosynthesis and utilizes the plastids from its food in a long-term way because itself, as an animal, has some of the genes in its nuclear genome that are required for photosynthesis. By any reasonable standard, that is macroevolution. And again, This is directly observed. We can see this happening in nature right now. We've also generated new things and observed the generation of new things, like new genetic traits or new proteins or new biochemical functions in nature. So we have nylonases, which are a class of enzymes that degrade nylon, a synthetic fiber. They're adapted from an earlier set of enzymes, but they are the nylonases that target the artificial fiber are new. Those don't exist prior to, like, there's no nylon-eating bacteria before nylon existed. We also have completely de novo antifreeze proteins in Arctic fish, and there's a gene in HIV called VPU, and that gene acquired a completely new function as part of the shift from infecting chimps over to infecting humans, a completely new biochemical function that required multiple mutations and novel protein-protein binding sites. So these are all new biochemical traits that we've directly observed. And then we have a whole category of things that we haven't necessarily directly observed occurring, but they're required in the young Earth model to have occurred in the recent past. One of these, my favorite one of these, is lizards, not just giving live birth, but having actual placentas. There are some lizards that not only give live birth, but they have placentas. Now, in the young Earth created kind model, those lizards are going to share a created kind because it's only specific species that do this. They're going to share a created kind with many other lizards, some of which may give live birth through a different mechanism called oviviviparity, where the eggs kind of develop internally and maybe lost their shell, but it's still kind of egg-based development, but it's internal. And they might just be oviparous. They might just lay eggs. So you've got this gradient of different reproductive modes among lizards that are in the same created kind, according to organizations like AIG. So you can't say that these large-scale, quote-unquote, macroevolutionary changes don't happen. We've directly observed them. They are necessary for the young Earth model in some cases, unless you want to reduce kinds down to the species level. Okay, so that's the point there. We could say one kind has never been observed, changing into a new kind with new information. That's wrong, unless you think evolving a placenta is not a change in kind, but I think that constitutes a change in kind. And I am following the comments here, so thank you, everybody. Hi. I do want to comment. Actually, hang on one second. Here we go. I just want to pull this up because endosymbiosis is actually not creating new kinds. It's just a co-adaptation. No, endosymbiosis does result in new kinds because endosymbiosis is an irreducible trait according to the traditional definition from Michael Behe. In order to have endosymbiosis, you need to have a number of things happen simultaneously. You need the food to not get eaten. You need the host to both not digest the food, but also not eject the food. You also need the trafficking of proteins to the algae or bacteria that is consumed. An example I talked about with the Pollinella becoming a new type of green algae, that's a bacterium that's getting eaten. It's primary endosymbiosis. You need to traffic proteins to it to keep it alive as its genome reduces because it's living inside you and stealing some of your food, so it loses genes. So you need to traffic things to it to keep it alive, but also you need to traffic some of its genes into your nucleus and then send those products back in order to keep it alive. If all of these things don't work, if all of these interactions don't work exactly right, then that relationship falls apart. Right? So that is a novel irreducible trait. The presence of an endosymbiotic organelle is a novel irreducible trait. And the transition from a heterotrophic amoeba-like organism to a photoautotroph with a new type of primary plastid, that is unquestionably a novel trait and an example of macroevolution. So I really like that example. So they go on to say, without a way to increase information, natural selection will not work as a mechanism for evolution. Evolution would agree with this. Yes, we do agree with that. Natural selection doesn't do everything. There's other stuff in there. But they believe that mutations somehow provide the new information for natural selection to act upon. It's mutations, recombinate, and there's other mechanisms. It's not just mutations, but we're going to focus on mutations in this chapter. So let's do it, right? Let's talk about mutations. Can mutations produce new information? I love this. Actually, scientists now know that the answer is no. There are tons and tons of examples of mutations producing new information. Now, the first thing we have to consider here is that there's no definition of information given here. In the simplest sense, right, when you have a mutation that occurs and you have a new allele, a new version of a gene that appears in a population as a result of that mutation, in the simplest sense, that is new information. When a gene duplicates, you've increased the amount of stuff in the genome. In the simplest sense, that is an increase in information. But what creationists normally mean here is that you don't see an increase in information of like functional information. You don't get like new interactions, new binding sites, new biochemical activity, whatever. That's normally what creationists mean here. Because if you define information in the simplest way, then it's trivially easy to show that mutations and other evolutionary mechanisms increase information content. Turns out it's easy to show that information increases when you define it more strictly as well, as we'll get to. So this is just silly. We have lots of examples of, like I told you, an example, a few examples of new genes just a few minutes ago. That's new information, right? So you can have a gene duplicates, and then one of them specializes, do something different. That's new information. You have just a non-coding region that becomes a coding region, happens relatively frequently. That's new information. This is trivially easy to document, and we've done so. So, hang on, we got to go back. I went too far. Sorry. Okay. In this chapter, I'll bring up several examples of evolution, particularly mutations, and show that information has not increased. But in all the reading I've done in the life science literature, I've never found a mutation that added information. See, it just doesn't, either that's a lie or the author here, which is Ken Ham and Jason Lyle, they don't read enough because there's lots of examples of mutations generating new information. I've provided some of those examples here. So this is another just blatant misstatement. All point mutations that have been studied on the molecular level turn out to reduce the genetic information and not to increase it. That's not true. One reason it's not true is because most mutations don't have any effect on the organism. They're in a neutral region, or if they are in a gene, they may be in a synonymous site where they don't change the actual protein structure, the amino acid sequence. Or maybe they do change something, but it's a neutral change. Most mutations are neutral. So you can find plenty of mutations that don't reduce genetic information. It's just silly. Plus, if you have a harmful mutation that breaks something, the reversal necessarily increases information. That can happen. So, like, there you go. So, for example, if the sickle cell, they'll say, not in this chapter, but creationists in general will say, oh, the sickle cell mutation, sure, it makes resistant to malaria, but that breaks a gene. That's a loss of information. Okay, what if it reverses back to the wild type? That's a gain. That's an information gaining mutation. Easy. So, like, clearly such mutations exist. The next line here is a problem. The NDT, neo-Darwinian theory, is supposed to explain. Stop right there. Stop right there. The neo-Darwinian theory is from the 1940s. That's the modern synthesis of evolution. We've done a lot of work since then. Things like neutral theory, endosymbiotic theory, niche construction, epigenetics, all kinds of advances in genomics, evo-devo, right? That's all after neo-Darwinism. So to argue against neo-Darwinism, either you're deliberately, kind of misstating the state of the field of evolutionary biology, or you don't know the current state of the field of evolutionary biology, and you actually think that neo-Darwinism is the current state of the field, which is not the case. Let's keep going. So there's more pages in this chapter. Okay, the failure to observe, this is silly. The failure to observe even one mutation that adds information is more than just a failure to find support for theory. It's evidence against theory, but that's wrong. That's not true. That's not true. Also, a thing I forgot to mention is that the source for the all-point mutations that reduce genetic information, the source from that's from 1997. That's from before we sequenced the human genome. I just want to point that out because, like, be up to date in your research, please. This book was from 2006, so it's less out of date than it is now, but like, that's still really out of date. That's really out of date. Should be using up-to-date research here. Okay. Oh, yep, 747. You know what's in there. Okay. All the parts of a 747 need to be assembled before it can fly. All the parts of biochemical machines in a cell need to be in place or it can't function. There's a phrase some of you are thinking right now, and you're 100% right. And if you read down here, Dr. Michael B., you know what's coming. Associate Professor Biochemistry describes these biochemical machines as examples of irreducible complexity. My favorite. It's an oldie but a goodie, irreducible complexity. There are 3 ways to interpret that concept, and they're all wrong. The first interpretation is the idea that if you read the plain text of Behe's definition, it only includes natural selection and mutation, not any other evolutionary mechanism. So like it's just irrelevant to the question of like, can something evolve? But assuming he means that it applies to all evolutionary mechanisms, you have 2 options. You could have the strong version where like nothing that has irreducible complexity can evolve, period. Anything that meets the criteria can evolve, the end. That version of the hypothesis is false because we've directly observed irreducibly complex systems evolving. That HIV protein, VPU, that I mentioned a few minutes ago, that's an irreducibly complex trait. It requires multiple mutations and leading to multiple novel protein-protein interactions. And you need to have all of those things present in order for that new trait. And that new trait is required for the jump from chimps to humans, for that virus to survive in humans. That's an irreducible trait. And we know it evolved. Like that thing definitely evolved. The Lenski-Sit plus trait, which was evolved in the lab in an experiment, in the long-term evolution experiment, that's also an irreducible trait that we directly observed evolving. The third version of the irreducible complexity hypothesis is the what I call the weak version. And the weak version is that, well, some things that have irreducible complexity can evolve and some things can't. That's an unfalsifiable game of whack-a-mole, because what that really means is we've explained how some systems had evolved and we haven't explained how other systems have evolved. And you could point to any of the systems in that latter group and say, irreducible complex, they can evolve, right? When the reality is we just haven't yet explained it in a step-by-step way to Michael Behe's satisfaction, right? So that's unfalsifiable, so we can dismiss that out of hand because it's not actually a testable hypothesis. So irreducible complexity, as always, is invalid argument. Sorry, I have to just 1 second, everybody. Okay, so our next thing here, our next page is, see, I keep skipping. Here we go. Okay, here we go. At the cellular level, thousands of machines are needed before life is even possible. So now we are talking about the origins of life as irreducibly complex. And the assumption here is that the simplest systems that exist right now are representative of the first living systems to exist on Earth, and that is a flawed assumption. The first living systems were undoubtedly less complex than the simplest cells that exist right now. So this assertion right here, not only does life require a source of information, but the complex machines must be in existence right from the start. That is false. That is wrong, because the earliest forms of life would not be represented by the most complex forms of life that we see, or the simplest forms of cellular life that we see on Earth right now. Continuing on, we have some random word generating things, but none of that's actually relevant. So here, the other complication is that the language system that reads the order of the molecules in the DNA is itself specified by the DNA. So we've got a chicken and egg problem, right? No, we don't. No, we don't. This is another one of those machines that must already be in existence, fully formed for life to work. That's not what the original hereditary system would have been. We know this. This is not complicated. Presumably the authors here know about the RNA world hypothesis and the idea that a combination of RNA molecules and peptides would have been the original system, not a DNA-based system, right? So that's the false information that's right here. We've already shown that information cannot come from mutations. That's not true. That's just a lie. One thing we know for sure from science, that information cannot arise from disorder by chance. It always takes greater information to produce information, and ultimately information is the result of intelligence. This is completely wrong, and I've cited a number of experiments that have completely disproven this statement. My two favorites are the paper on random polymerization. You incorporate random either RNA sequences or polypeptides into cells, and it has some beneficial phenotype. And the lac promoter study, where they took the promoter for the lac operon, which is a gene expression module in bacteria. They removed the wild type promoter, and they put in there random bases of the same length. It's about 100 nucleotides. They put random sequences of the same length in its place, and those random sequences were able to operate as a lac promoter. So down here, there's no known natural law through which matter can give rise to information. Neither is any physical process or material phenomenon known that can do this. So natural law, that's like irrelevant. Who cares? But there are processes that can do this. We've documented that this is the case. You can get information from non-directed processes, from probabilistic process, or if you want to use the word, random processes. That's a thing we have demonstrated. Okay, so what's the source? What's the source of information? We can conclude that huge amount of information must originally come from intelligence. Nope. Can't conclude that. We've documented that you don't need intelligence to get information. It's just a lie to say otherwise. The end. So let's keep going through this. So now it gets into a lot of religious stuff, and I don't have anything to say about that. That may actually be the end here. Here we go. Some people claim that it's unscientific to start from God's word. And it can be, but it doesn't have to be, right? Because you could say, okay, we're going to start with the Bible, and we're going to use that as the basis for hypothesis testing, right? Okay, the Bible says X, therefore we can predict blah, blah, blah. And we can then go out and test that, right? So like, that's fine. You can do that, but you can't start with the Bible, for example, as an unassailable premise in your argument and never allow yourself to deviate from that. That's unscientific, but it's perfectly reasonable to start with the Bible and use that as the basis for hypothesis testing. That's something we can do. Right. Now, there's a lot of stuff I skipped in here about like man's word versus God's word. AIG and Ken Ham love that stuff. I don't care. I'm leaving all that stuff out. It doesn't matter to me. Okay, so now we get into like some religion-y stuff, and it doesn't matter, but there's a little bit right at the end here that I want to just point out, and it's this. Over and over again, the Bible's historical account has been confirmed by, and I'm skipping archaeology, geology, and astronomy because those aren't my fields, but biology. That's not true. That like we can make predictions, as I just described, based on like a literalist interpretation of the Ark story, for example, and make predictions about genetic similarity and convergence in the past. And it doesn't hold up, right? It actually contradicts the literalist interpretation of those stories in the Bible. No contradiction or erroneous information has ever. Yeah, it's not true. It's just like we can hypothesis test based on this stuff and show that it's not standing up to scrutiny. So I think that takes us to the end of the chapter here. Yep, that's the end of the chapter. Now we're up to the references. So that's chapter 1. This chapter started off with a whole bunch of theology and philosophy stuff, but it got really into biology, specifically design and detecting design in biological systems and the sources of design and information. And in those specific contexts, which are the contexts I'm going to be commenting on throughout this series, in those specific contexts, almost none of the statements they made as like factual assertions were actually accurate. And again, I've only read a few chapters of this book, but I anticipate that all 4 books are going to be kind of like that based on what I've read so far. So that is the end of part 1 in this series on the New Answers books from Answers in Genesis. Join me next week for part 2, where we will continue with this. And one more note I will say here. is that it is very much, I'm just going to pull this comment up here because this is exactly right. Yeah, these books so far, to the extent that I've read them, it's extremely repetitive, old hat stuff. If you're familiar with the evolution and creationism discourse, you're going to recognize these arguments, and you're often going to know why they're not good. So play along with me as we go through these books and try to figure out, before I say it, what the problems are. Thank you all for watching. I hope you all have a great night, and I will see you next week for part 2 in this series. Good night.